So these are the phage progenies with the cosmid vectors within that. This is the cosmid vector within that phage head. Then after the formation, these phage progenies, uh, you can use to infect these phages with another bacterial cell. Then these phages may introduce or insert this vector into that bacterial cell, and within that bacterial cell, these cosmid vector may get circularized. The copies of that cosmid vectors may be formed within that. So for the construction of a cosmid vector, it required different elements. DNA elements need to be introduced into the vector. The first and foremost element required to be introduced into that cosmid vector is that of a cos sequence. The cos sequence that had been obtained from a phage, lambda phage. And the second important feature to be incorporated into this vector is an origin of replication. We have discussed in detail about that. You can introduce different kinds of origin of replication into it. On accordance with that, you can maintain this cosmid vector into different types of cells. I have told you that if you have introduced SV40 origin of replication, then you can maintain them in mammalian cells. Likewise, you can try different original applications. And the third feature to be incorporated is a selectable marker. See here in this picture, the selectable marker is shown as a ampicillin resistant gene. So there are different selectable markers out there. In different cosmic vectors, you can introduce any one of it. These selectable markers help us to select the transformant bacteria after the introduction or the transformation of this vector into bacteria. And another feature to be introduced into phospholipid vector is multiple cloning sites or different restriction sites into it. Hence, you can introduce different foreign DNA, is cosmid vectors.